Hello, this is Dejon Hill. I was assigned the motor proteins, myosin, to discuss, which is what I'll be explaining here in this PowerPoint presentation. As far as they go, they are composed of many alpha helices. I'll go into discussion later in terms of different sections as far as their different responsibilities to ensure that the protein is functioning properly. Myosin is abundant all throughout the body. The main one that I'll be talking about here today is myosin 2, which is primarily actually in skeletal muscle cells, but it does have the other myosin are actually present all throughout the body. And then you should know that myosin is responsible for the movement in all organisms. That goes from me like lifting my arm right now to the way that amoeba kind of moves like a squiggly, has a squiggly movement going on around the outside of it. Myosin is responsible for all of that. Here is an image here going from large to small here. Of course, we have muscles in our bodies, and then here you see this represents one muscle fiber or cell, and inside of that is a strand of myofibrils, which is composed of these units here, which are called sarcomeres, and these units are obviously repeating over and over until it's large enough to form a full muscle fiber or cell. Excuse me. But this is just showing a basic um, layout as far as where myosin is within the body. You see here these zigzag lines are what are known as the Z disc. And here, right in the middle, are the areas known as the M line, which is basically just kind of like an open area where there are no myosin heads. The myosin heads react here with the actin filaments to pull those Z discs in closer, and that is how we get a muscle contraction. Now, as far as the way myosin looks, we see that it's composed of these head regions, and then there's a neck region like here and here, and then after that is a repeated tail sequence. Again, all of these are alpha helices, as made evident by this ribbon model here, and each of them have their own particular functions. Like in this case, you see these light chains, which are represented here, and then here, and here. And those are responsible for providing stability for myosin to perform the way it should be as far as causing muscle contractions. And here you have the actin binding domain, which is what's bound to the actin filament, as I showed here. It's bound here. These points that touch represent the actin binding domain. And that part of the myosin protein undergoes some conformational changes once ATP uh, binds to the head there, and then it causes some changes that eventually lead to a muscle contraction, which I'll go into more detail about here in just a second. As previously discussed, here now is represented as a straight line. The Z disc gets pulled in. Each of them get pulled in closer whenever ATP is attached and activates the myosin protein to do its part. The way this works, I'm not sure if this picture makes it very clear or not, but neither of these portions changes in length at all. The myosin stays one length, and then each of these actin filaments stay one length also. The way that a contraction occurs is that each of these myosin proteins, the head region walks, like I said, but the amount of time that it's taken for me to explain that is not nearly as long as it takes for them to essentially walk from this point to there. So it happens rather quickly, and that's again after ATP gets in there and activates this protein, and also the presence of this calcium ion causes some movements there. So I'll explain the muscle contraction here in terms of this video. So it's a lot going on there all at once, but I'll try to explain everything here, and hopefully it's represented there visually easily. So we see here this yellow particle. It keeps repeating over and over. So we see this yellow particle coming down and attaching here to this, I guess, pink colored section. That represents troponin. And these blue lines attached to that particular, to that part right there is called tropomyosin. And what's happening here is that in order for the myosin protein to attach here to this orange coil, actin, it first has to have access to its binding site, to the actin binding site. So we see that the calcium comes in and it attaches to the troponin part there, which causes the troponin to move out of the way of the myosin protein, and this green large structure that keeps attaching over and over to what's obviously the myosin protein here is ATP. Now this gray cone keeps coming in, it keeps poking it. That is a representative of magnesium, which comes and actually activates the ATPase enzyme that's present in the myosin protein. And once that happens, then you see now it goes from one big green ball to two smaller particles being shot away from the myosin there in this illustration. And what that is a representative of the ADP and phosphate that's released as a result of all this. So this is the walking that I mentioned was going on there. This particular illustration doesn't quite show the myosin head region as it undergoes changes, but this whole time it does change. It goes from whatever shape it's in while it's bound to the actin filament to a different shape while it has this ATP attached to it. And then once the magnesium comes and activates the ATPase, hydrolysis occurs, and that's why the ATP is split then into ADP and phosphate. And once they're released, then myosin will go back to the shape that enabled it to attach to the actin filament there in the first place. And so as far as myosin goes, there are some diseases that are associated with issues as far as this protein goes. And like I said, here it's due to mutation of amino acid sequences. In some cases, you can have what's called hypertrophic cardiomyopathy, which is a muscle thickening of the heart. It's a mutation that causes this, as far as this particular process here, it has too much force as far as this particular process goes, and that's what causes the muscles to be thicker, and it also causes it to grow larger. And then you have also dilated cardiomyopathy, which is a weakening of the same heart muscles, and that would be due to weak interactions occurring between myosin and the actin filament, as previously displayed there. So for hypertrophic cardiomyopathy, is too much force, and dilated cardiomyopathy, not enough force. And then there's also congenital myopathy, which is basically any birth defect that's caused, any muscle defect that a person may have from birth. So that just about wraps up myosin. Thank you.